Okay, let's dive into something truly incredible. The sheer complexity of our immune system, and specifically, the intricate journey of B cells.
Yeah.
Those amazing antibody factories. They have this monumental task navigating our bodies, finding the right places, and deciding exactly when and where to jump into action.
Mm.
It's a ridiculously delicate dance, and fine-tuning it is absolutely crucial, not just for fighting off infections, but also for preventing our own immune system from, well, mistakenly attacking us in autoimmune diseases.
That precision is everything. It's not enough for a B cell to just recognize something potentially harmful.
Yeah.
It has to get to the right microenvironment, interact correctly with other cells, and then activate only when appropriate.
Yeah.
Understanding the molecular choreography behind that navigation and response is fundamental.
Exactly. And that brings us to the source material for this deep dive. We're unpacking a fascinating scientific paper titled Galectin-9 Bridges Human B Cells to Vascular Endothelium While Programming Regulatory Pathways from the Journal of Autoimmunity. This is your source, your specific questions about this research.
Our mission is to pull apart this paper, focus on a key player in this complex immune interaction, a molecule called Galectin-9, or Gal9 for short,
Yeah.
and figure out what this particular study reveals about its surprising roles with B cells, how it impacts immunity, and what this might mean for future therapies.
So giving us a bit of context from the paper's introduction, what was the general understanding of Gal9 and B cells before this research came out?
Right. So it was known that for humoral immunity, which is driven by antibodies produced by B cells, those B cells need to get into lymphoid organs like lymph nodes.
Mm-hmm.
Gal9 was already identified as a molecule that could bind to naive in memory B cells.
Yeah.
Previous work suggested it acted as a negative regulator, meaning it could, put the brakes on their B cell receptor, or BCR signaling. That's the primary way B cells sense their environment and potential threats.
So kind of a known off switch then.
Pretty much,
Yeah.
yeah. And mouse studies had supported this idea. For example, mice deficient in Gal9 showed increased B cell activity. Conversely, treating mice with Gal9 could suppress antibody responses in models of vaccination or even reduce disease severity in a lupus model.
Oh.
So the regulatory potential of Gal9 was established, but exactly how it exerted this control, especially spatially, within a lymph node, that wasn't fully understood.
Which leads us right into one of the first major twists this paper revealed. Okay, let's unpack this. The unexpected location of Galectin-9. Where did they actually find it when they looked closely?
This was one of the initial surprises that really changed the perspective. They looked at human lymphoid organs, like tonsils and lymph nodes, using techniques like immunohistochemistry and immunofluorescence, essentially, staining to see where specific molecules are.
Right.
While they confirmed Gal9 on B cells, they found it was incredibly abundant, conspicuously high, not just on the B cells, but lining the blood vessels within these tissues.
Wait, so it's on the walls of the vessels, not just floating around or on the cells themselves?
Exactly. Specifically, they saw high levels on the endothelial cells. That's the inner lining of high endothelial venules, or HEVs, and postcapillary venules.
Okay. HEVs.
Yeah, HEVs are actually specialized blood vessels in lymph nodes that act as key entry points for lymphocytes. Figure 1 in the paper really highlights this, showing clear co-localization of Gal9 staining with markers for these HEVs, distinct from the B cell staining.
Wow.
They even back this up by referencing public mouse data showing high Gal9 in lymph node stromal cells, including the endothelium.
Okay, that's a significant finding. So this molecule, known to interact with B cells in a regulatory way, is highly expressed right where B cells might be trying to enter or leave the lymph node. What's the implication of finding such high amounts of Gal9 in that specific vascular location?
Well, it immediately suggested a potential physical interaction, a spatial role. If B cells have molecules on their surface that bind to Gal9, which they do, and Galang is plastered all over the highway of the lymph node blood vessels, it strongly implies that Gal9 might be involved in how B cells stick to or move across those vessels.
Which naturally brings us to the next big question. Galectin-9's role in B cell movement and adhesion. If it's on the vessels and B cells bind it, what happens when they connect? Does it affect their ability to stick or move?
That's precisely what they investigated next. First, they characterized the adhesion properties of human circulating B cells. What they observed, shown in Figure 2, is that human circulating B cells have high expression of Gal9 ligands, the carbohydrate structures Gal9 binds to.
Okay.
But interestingly, compared to T cells, which are quite good at sticking to vessels via classic adhesion molecules like L-selectin, Human B cells express relatively low levels of these standard molecules.
Yeah. So based on the sources, how human B cells initially stick to vascular endothelial cells wasn't as clearly defined as it is for T cells.
That's right. This paper provides compelling evidence for an alternative or, complementary mechanism. And the adhesion results they got were very clear. Using a model system with human vascular endothelial cells, specifically HUVECs, which are human umbilical vein endothelial cells commonly used in lab studies, they demonstrated that adding Gal9 significantly promotes human B-cell adhesion.
How much of an effect are we talking about? A big difference.
It was a substantial increase, yeah. In static assays, where cells are simply layered on the surface, they saw a 2.5-fold increase in B-cell sticking.
Wow.
Even under conditions simulating blood flow, known as shear flow, they observed a 2-fold increase in shear resistance, meaning the B cells were holding on much tighter to the vessel lining. Figure 4 shows this data.
Okay.
Crucially, this effect was specific to Gal9. Other related molecules like Galectin-9 or NECA3 didn't work, and it was inhibited by lactose, confirming it relies on Gal9 binding to its carbohydrate ligands.
Right, the sugar binding.
Exactly. They also found that stimulating the HUVACs with interferons, which happens during inflammation, increased Gal9 expression on the ECs and boosted B-cell adhesion, reinforcing the potential relevance in physiological contexts. That's in figures 3 and 4.
Okay, so Gal9 on the vessel wall definitely makes B cells stickier. But what about the next step, actually getting through the vessel wall, migrating into the tissue like a lymph node?
That's where it gets even more fascinating. In experiments measuring transendothelial migration, or TEM, where B cells have to cross a layer of endothelial cells, Gal9 actually reduced migration by about 40%. That's figure 5A.
Reduced migration, so it helps them stick, but then hinders their ability to cross. That seems counterintuitive.
Exactly. It seems to make them adhere more strongly while simultaneously slowing down their movement through the vessel lining. They also found a potential molecular clue here. The reduced migration was linked to reduced phosphorylation of FAK, or forkyl adhesion kinase. That's figure 5B.
FAK.
Yeah, FAK is a key protein that cells use to sense and interact with their environment, and it's heavily involved in cell movement.
That's a critical detail. So, putting these findings together, the study suggests Gal9 on the vessel acts like a kind of bridge, right? It promotes initial adhesion, but then potentially puts the brakes on the crossing process.
That's right. It makes them stick and slows them down.
Yeah.
This could significantly influence how and when B cells enter or exit tissues like lymph nodes. And it suggests B cells might use different or, additional mechanisms for vessel interaction compared to the classic L-selectin-mediated homing story that's been well established for T cells, which is something the paper discusses.
That is fascinating. So it's not just about finding the door, it's about how you interact with the door frame. Now, what happens inside the B cell while it's potentially stuck or slowed down by this Gal9 interaction on the vessel wall? Let's shift focus to Galectin-9's influence on B cell programming and signaling. Does simply binding to extracellular Gal9 change the B cell's internal instructions?
It absolutely does. They performed RNA sequencing on naive B cells that had bound to Gal9. This is a technique to see which genes are being turned on or off.
Right, looking at the messages.
Exactly. And they saw a distinct and specific gene expression program being initiated. You can see that in supplementary figures 3 and 4.
What kind of program, what themes emerged from looking at all those gene changes?
Well, the gene ontology analysis, which groups genes by their function, like in supplementary figure 5, highlighted some prominent themes. There was a conspicuous upregulation of pathways associated with dampening cell activation, improving cell survival, and stress-activated signaling.
Okay, dampening activation.
Yeah, and at the same time, there was a noticeable downregulation of pathways involved in membrane and cytoskeletal dynamics, things related to how the cell moves and changes shape, and also protein modification.
Okay, so Gal9 binding seems to send signals that tell the B cell, stick around, stay alive, but don't get too excited or prepare to move much. Is that fair?
That's a great way to summarize it. It seems to promote a state of quiescence or stasis while maintaining vitality. It's like putting the cell in a holding pattern, potentially right there on the vessel wall.
Were there any specific genes that really stood out in this reprogramming? Any key players?
Yes, they focused on several immunoregulatory genes that were clearly modulated, and one that was particularly interesting was SLAMF7, also known as CS1 or CD319.
SLAMF7, I've heard of that.
Yeah, this molecule is known to be involved in signaling in various lymphocyte types, and is actually a target for therapies in multiple myeloma.
Okay, so SLAMF7 is upregulated. Does that mean this signal is getting the B cell ready to activate? Because you said activation pathways were dampened.
Here's where it gets really interesting and quite clever.
Hmm.
SLAMF7's effect, whether it promotes activation or inhibition, depends critically on which adapter protein it associates with inside the cell. One key partner is ET2.
ET2, okay.
When SLAMF7 teams up with ET2, it enhances cell activation. But this study found something remarkable. Gal9 binding simultaneously caused a clear increase in SLAMF7 mRNA and protein levels, that's figure 6, while strikingly causing a dramatic decrease in ET2 mRNA and protein levels in naive B cells.
Mm-hmm.
We're talking 60–75% down, shown in figure 7A and B.
Wow. Hold on. So it gives the cell more of a receptor, SLAMF7, that could enhance activation, but then it removes the essential partner, ET2, required to make that happen.
Precisely. The dramatic downregulation of E2 means that even though SLAMF7 levels go up, in the absence of its required activating partner, the upregulated SLAMF7 is now effectively configured to deliver inhibitory signals instead of activating ones.
That's a really sophisticated molecular switch.
It really is.
And you mentioned survival signals, too. Was that separate?
Yes. Alongside this SLAM7 ET2 dynamic, Gal9 binding also caused an increase in the phosphorylation of ERK, or PERK. That's in figure 7 as well. Phosphorelated ERK is a well-known pro-survival factor in many cell types, including B cells. The paper's discussion also noted that ERK is linked to adhesion pathways, potentially connecting these different effects.
Okay, let's try and connect the dots from all this.
Yeah.
It's complex, but the picture emerging is that Gal9 on the vessel wall isn't just a physical sticky patch. It's making B cells adhere, slowing down their crossing.
Right.
And simultaneously, through the act of binding the extracellular Gal9, sending signals into the B cell that promote survival and basically tell it to stand down, preventing activation via this clever trick with SLAMF7 and ET2, among potentially other pathways.
That's the synthesis presented in the paper, yeah. It's illustrated conceptually in figure 8. Gal9 on the LN vessels physically bridges B cells to the endothelial cells, impacting how they move across. While this is happening, the extracellular Gal9 binding triggers intracellular signals that promote cell survival and impose that state of quiescence or stasis by activating immunoregulatory pathways and suppressing genes involved in cell dynamics.
And they propose this overall inhibitory effect could be happening through multiple potentially reinforcing pathways, not just the SLAMF7 thing.
Yes. They discuss the previously known interaction of Gal9 with the phosphatase CD45, which can influence signaling downstream via the Lin-CD22-SHP1 axis. That was kind of the known pathway.
Okay.
And this new study reveals the parallel or perhaps synergistic SLAMF7-mediated inhibition because ET2 is downregulated. Seems to be a multi-pronged regulatory action.
So what does all this mean for you listening right now? Why should you care about Gal9 making B cells stick to blood vessels and playing molecular tricks with SLAMF7 and ET2?
Because these fundamental activities likely play a crucial role in maintaining B cell homeostasis, that critical balance in the immune system. By regulating B cell entry, exit, and activity right at the vascular interface, this mechanism could be vital for controlling potentially autoreactive B cells, the ones that might cause autoimmune disease in the periphery, perhaps before they even cause trouble.
Like catching them at the gate.
Exactly. As the abstract and discussion highlight, this could be a key aspect of what's called peripheral tolerance.
And the prior mouse studies you mentioned earlier where manipulating Gal9 affected B cell responses, they fit perfectly with this idea then.
They absolutely do. Gal9 deficiency in mice, leading to increased germinal center size and antibody responses, makes sense if Gal9 is normally putting a brake on B cell activity.
Right.
And Gal9 treatment, inhibiting vaccine responses or ameliorating lupus models, aligns with Gal9 acting as a regulator that dampens B cell immunity. It all reinforces the idea that Gal9 is a significant and perhaps surprisingly located regulator of D cell behavior and function.
That's a lot to process, but it really lays bare just how intricate the immune system is, right down to the molecular details of a cell, deciding whether and how to interact with the blood vessel wall. So let's pull out some of the big aha moments from this deep dive.
For me, the first big surprise was definitely finding Gal9 in such high amounts, specifically on the blood vessels within lymph nodes, rather than primarily just floating or on the B cells themselves.
All right, the location was key. And realizing that this vascular Gal9 isn't passive, it actively acts as a bridge, making B cells stickier to the vessel wall and, surprisingly, slowing down their ability to cross through it.
Exactly. And then the really profound finding that this extracellular Gal9 binding simultaneously reprograms the B cells internally, promoting their survival but imposing a state of quiescence or stasis.
Keeping them alive but quiet.
Mhm.
And the elegant mechanism behind that reprogramming, specifically how it increases SLAMF7 but then removes its necessary activating partner, EET2, essentially reconfiguring SLAMF7 to deliver an inhibitory signal instead. That's quite a trick.
It's a beautiful piece of molecular regulation, highlighting the complexity and maybe the redundancy built into the system.
And the overall relevance, just to bring it home.
This study points to a fundamental process for how the immune system might be controlling B-cell activity and movement, especially in peripheral tissues like lymph nodes. This could be crucial for maintaining immune balance and preventing unwanted responses like those seen in autoimmunity.
Which brings us to the potential future. If we understand this pathway involving Gal9 and its interaction with B cells and their ligands, could this be something we could potentially manipulate therapeutically?
Absolutely. Understanding this intricate interaction opens up potential strategies. You could imagine trying to boost antibody responses, perhaps to enhance vaccine efficacy by blocking this inhibitory pathway. Or, conversely, trying to blunt unwanted autoimmune responses by mimicking or enhancing this Gal9-mediated inhibition. It offers potential targets for intervention.
So here's a provocative thought to leave you with, based on this research. If manipulating Gal9 could effectively dial B-cell activity up or down, making them stick more, cross less, or be less prone to activation, how precise could we really get with that?
Yeah.
Could we target specific B-cell subsets or contexts for, say, a vaccine boost without inadvertently running the risk of dampening overall immunity or even triggering unintended autoimmune consequences somewhere else?
Yes. That's the million-dollar question, isn't it? It highlights the incredibly delicate balance the immune system maintains. Unpacking fundamental molecular interactions like those involving Gal9 reveals both immense potential for new therapies and, well, the complex challenges involved in safely harnessing that potential without disrupting the system's core harmony.
A lot to think about. Thanks for taking this deep dive with us into the world of Galectin-9 and B-cell regulation.